Hello, everyone. Welcome to The Current. This is the North Central Region Water Network's Speed Networking webinar series. And welcome to this webinar and welcome to 2022. Happy New Year, everyone. I am Rebecca Power. I'm joining you from the University of Wisconsin-Madison Division of Extension. I don't usually wear the gear, but I've got the gear on today. Since some of you may be new to the North Central Region Water Network, we are an extension-led collaboration among 12 universities and our partners in the upper Midwest. So we are so pleased to bring you this webinar today, which is about microplastics and the impact of microplastics in water, particularly freshwater systems. So I think microplastics have been emerging and have now emerged as part of our challenges with water quality and all the associated impacts of plastics in our environment. So today we are pleased to have two speakers talking about different aspects of microplastic. First, Sherry Sam Mason, Director of Sustainability at Penn State Erie in the Barron College, and Melissa Daum, Assistant Professor of in the Department of Ecology and Evolutionary Biology at the University of Michigan. So how this session works, we will ask you to please submit your questions for our presenters in the Q&A panel, the Zoom Q&A panel. We will have a dedicated Q&A session following both presenters' presentations, and you should be able to find that Q&A panel at the bottom of your screen. Please look at the questions that other participants are submitting, so you can upvote the ones you are most excited to hear about yourself, and we'll go ahead and ask those questions at the end of our session. If you are experiencing any technical issues, sound issues, or have other types of questions that aren't related to our presenters, you can use the chat feature for that, and the chat feature is similarly located at an icon at the bottom of our webinar screen. If you are having audio issues through the web interface, there is a phone-in option, and that can be accessed by clicking the up arrow on the mute icon or the microphone icon and clicking switch to phone audio. We do record these sessions and post them at northcentralwater.org. We also live tweet these sessions, so you can join in that way as well. All right, I think those are all of our logistical details here. See if I can, there we go. Okay, so here again are today's presenters. You get the words as well as my saying them, and why don't we go ahead and get started with our first presenter, Sam Mason. So you can read a little bit more about her here. She has a bachelor's degree from the University of Texas at Austin, completed her doctorate in chemistry at the University of Montana, and was selected as an EPA environmental champion in 2016 and has various different awards that she has listed there. So she comes highly recommended. So with that, I will go ahead and stop sharing my screen, and we will turn it over to Sam. And Sam, you are muted still.
I am. Thank you. Thank you for having me. Thank you all for being here. And hopefully you can see my screen pretty well now.
Perfect.
Perfect. All right. So I've been tasked with the wonderful introduction to the topic. So plastic, though it's a material that is actually created at the dawn of the last century, really as a way to replace natural materials such as ivory that was being used for billiard balls. But it wasn't until the infrastructure for the mass production of plastics wasn't actually put into place really until World War II. And since World War II, you see this exponential increase in plastics production. I like to mention the fact that, this material is relatively new. It's really only been a common part of our society for about 70 years. And yet we have already produced over 10 billion metric tons of this material, half of which has just been produced this century. So since 2000, more than half of that has been produced. And a big driver for this is actually has been marketing. It hasn't been consumer driven. It's really kind of been pushed in part as a way of using byproducts of the fossil fuel industry. Of this 10 billion metric tons, about 30% of it is still in use on a day-to-day basis, right? And probably many of us can think of examples of that, leaving about 70% of it to have led to waste that's associated with this plastics production. Of that, 11% has been recycled. More of it, 15%, has actually been incinerated, and leaving about 74% of this plastic waste that has been generated being discarded. But when you see that statistic, it's a little misleading because you would think that all of that is in maybe a landfill somewhere, but current information suggests that somewhere between 10 to 15% of it has actually ended up in the environment. So kind of, more than what has been recycled, on par with the amount that has been incinerated is actually out in our environment somewhere. When you see, titles like this, news articles talking about dolphins and whales being stranded with their stomachs filled with plastic, turtles with straws up their nose, you can really kind of understand that a focus of this work has really been on the world's oceans. This is also where this topic kind of started in terms of the awareness and the research. It really started in the world's oceans and the 5 oceanic gyres that exist that have all been shown to contain mass accumulations of anthropogenic debris, most of which is plastic. And there's no doubt, right, that these ocean gyres have these mass accumulations of plastic. We have lots and lots of data to support that understanding. But when we look upstream from the oceans, we realize that plastic is making its way into the oceans through our freshwater systems. And should you think that this last image was taken from somewhere in like Bangladesh, that's actually Erie, Pennsylvania, where I live. Just as a point showing how this material makes its way from land into our freshwater ecosystems and from those freshwaters into the oceans. So I do live along the shores of the largest freshwater ecosystem in the entire world, the Great Lakes, the Laurentian Great Lakes. I feel extremely fortunate to live here, and it is shown. It's been a really interesting kind of proving ground for looking and understanding plastic pollution within freshwater systems as opposed to the ocean. So over the last dozen years, we have been doing studies, and I say we, including my colleague Melissa, who's going to be one of my co-presenters today, showing that there are concentrations of plastics within the Great Lakes that are comparable and rival those in the world's oceans. And they roughly increase as we follow the flow of water from Lake Superior into Lake Huron, Lake Erie, and then Lake Ontario, and then on out the St. Lawrence River to the North Atlantic Ocean. So you see that those counts roughly increase with the exception of Lake Michigan, though at this point in time, Lake Michigan is our least studied of the 5 Great Lakes. And these numbers really indicate that we have somewhere between 1 billion and 5 billion pieces of plastic across the surface of each of our 5 Great Lakes at any point in time. So I should have, kind of mention this at the outset, these are all surface floating plastics. If we look down in the sediment, we actually tend to see higher concentrations than we do along the lake surface because of the fact that they do settle over time. And Melissa will be talking more about that in her presentation in terms of the biosphere or the plastosphere that exists on these particles. Most of these, 97% of these plastics that we find within the water are what we classify as microplastics, which by definition are particles that are less than 5 millimeters in size. 75% of them are actually even smaller than that. They're smaller than 1 millimeter in size. Some of these are what we call primary microplastics. That is, they were manufactured to be micro-sized. So pre-production plastic pellets, this is how plastics are shipped from a fossil fuel refinery to a point where they are manufactured into plastic products. Sometimes these are also referred to as nurdles, would be in this category, as well as the microbeads that were included in personal care products. But probably more than that, more microplastics are what we call secondary microplastics. These are things that break down or break off of a bigger plastic items, either through a photooxidative degradation pathway, such as the fragments in the bottom center of the screen, or microfibers that come off of our clothes and other synthetic materials during their use and washing. When we look upstream from the rivers, from the lakes into the rivers, we actually see that the concentrations of microplastics are even higher than what we see within the Great Lakes themselves. In some ways, that's not surprising because you're closer to the people who are the ultimate sources of the plastic, and there's been less water for dilution of those plastics as a consequence. Interestingly, in our studies of Great Lakes tributaries, we found no relationship between the concentrations of microplastics and the percent of wastewater treatment plant effluent that contributed to that river, though other studies have found kind of differing information. So there's still a lot of kind of questions out there. Our studies of wastewater treatment plants have shown that on average, a wastewater treatment plant is releasing about 4 million particles of plastic per day on average for each facility. And there are 15,000 wastewater treatment facilities across the United States. Of these particles, 60% of them are those fibers that come off of our clothing as we use and wash those materials. So that's kind of interesting. Along that same note, right? So, the stuff is making its way into its water. I wanted to make sure, especially in this audience, to talk about our studies looking at microplastics and human consumables. We've done 2 studies in this kind of sphere of research. The first looked at table salt as one of the materials, and we found that on average, each kilogram of table salt had 212 particles of plastic. We also looked at beer. And this was specifically beer that was brewed using Great Lakes water, the kind of inference being that if there is plastic in the Great Lakes, then maybe there's plastic in the beer. And what we found was that on average, there are 4 particles of plastic within each liter of beer that we looked at. And then we also looked at, did a global tap water assessment. So this was 159 samples of tap water that were collected across the globe. And we found that actually there was more plastic within tap water as compared to beer, on average 5.5 particles of plastic per liter of tap water. After this study, as a follow-up study, we decided to look at bottled water, and we found that on average in this global bottled water assessment, so we looked at a total of 259 individual bottles of water across 11 different brands coming from 19 different countries. We found on average 325 particles of plastic within each liter of bottled water. I should note that there was a significant difference in the types of plastic that we found. in the tap water, the beer, and the sea salt as compared to the bottled water. In the former, found a lot of fibers, but in bottled water, it was mostly fragments. And this really led us to the conclusion that it's the act of packaging the bottled water that is leading to most of the contamination within the bottled water itself. So it's the manufacturing of the bottled water that is leading to that contamination. So this work was cited and is part of these kind of review articles that have looked across other studies in terms of kind of understanding the prominence of microplastic in a human diet, and bottled water was by far the worst, beer coming in second. And then I wanted to notice here that the air is up here, and really kind of trying to drive the point home with this final slide, the ubiquity of microplastics, that it doesn't matter if we look, at the top of Mount Everest or the bottom of the Marianas Trench. Wherever we look, we find microplastics because they are just so prominent and prevalent and ubiquitous within our environment. And with that, I'm going to stop sharing and allow my colleague, and look forward to our Q&A at the end. Melissa, it's all yours.
Great, thank you, Sam. And I'm going to just share my screen for a minute before Melissa gets started here. Okay. There we go. And just to introduce Melissa, our second presenter, and thank you again, Sam. So Melissa Daum is studied microbiology at Cornell, and then did some globetrotting there over to Germany, and did a postdoc at the University of Arizona before landing back here in our very own Midwest at the University of Michigan, where she's currently an assistant professor. So you can look there at a little bit more of her research and her areas of emphasis, but she comes at this from a microbiology perspective. So she's going to talk to us a little bit about the what's happening in that tiny world that we can't see related to these microplastics, and what that means for us. So with that, I will stop sharing and look forward to your presentation, Melissa.
Great, thank you. We'll take it over. I'm gonna try it one more time. We just have the one window.
And just a reminder, while she's pulling her slides up, if you take a look at the questions that other folks have submitted, feel free to either ask your own or upvote ones that have already been asked, and we will get to your questions after Melissa's through.
Does this look good and sound good? Everything?
Yes.
All right, great. So thanks for the introduction, and thanks for the invitation and the audience. As you heard, I'm an environmental microbiologist. So the way I've thought about the implications of microplastic pollution is through the microbial lens. So in our lab, we think about microplastic interactions in our freshwater systems that we study. And here on this first slide, you can see some of our early SEM images. Oh my, yes, back in 2014, it's dated right on the image of the biofilms that were living on plastics that we pulled out of Lake Erie. And as a microbiologist, you can see that they are actually quite mature. So most of them, it took some time and removal of many layers, many layers of biological material to get back down to the plastic surface. And these early images really inspired for us some basic questions of what are the consequences of the presence of those microbes, given what we know of metabolic diversity and potential for microbes to. And I will share a bit about how we think about this problem. Well, I'll give them to you straight off the bat. Which I'll discuss a bit more. Which we study in the lab. We will read microbes, the base of mere ecosystem. And that, there we go. Central, for instance, oxygen, nitrogen, phosphorus, iron, pretty much any of them. But there's probably more interesting and more tangible in of those cycles with the plastic cycle, life cycles, which we think about in the lab. So yeah, they are essential for these earth processes, but we also know that they're really central to the functioning of animal and really any host, and whether it's animal or plant life. So microbes rule the world. Take home one, check. The next is this idea of the plastic microbiome, or it's also described in our field as the plastosphere. So that's considering the kind of liminal space around a given piece of plastic, from microns to millimeters, in space away from or outside of the plastic. That can be the biological, chemical features of that novel biome. And within that plastosphere, we study the microbes. And we've learned that the microbes that are found on, living on in biofilms, on plastic in natural settings, are distinct, so that they're novel, distinct assemblages of microbes that live on plastic. And what that looks like in terms of the type of data that we apply, not only, well, I think by morphology, one would argue they are not distinct. These are things that we can recognize, different types of diatoms, small bacterial cells. But the way that we look at these communities is by sequencing either marker genes or full genomes of the microbes that are there. And so on the right here, you can think of the matrix that we sample. So we either pull pieces of plastic out of the environment to study the microbes on them, or we filter water, kind of like making coffee. And then we're able to extract the DNA from the filter, sequence it, and generate a species list of microbes that are either living in the water, so that would be these free living microbes, living on presumably biogenic particles in the water, as well as those living on the pieces of plastic that we're able to pull out by hand. And so what you'll see on the left will be an ordination that essentially tells you how similar are the species lists of the microbial communities that we are able to identify and reconstruct from these three different biomes: water, plastic, and other particles. And they'll be distinguished by different colors. And what this tells us then, when we look at these hundreds of species lists, and these are all from Lake Erie samples, where we stopped at a number of different sites, pulled out plastic, filtered water, we're able to see that the community composition of the microbes that live on the plastic was overwhelmingly, significantly distinct from those that we see in the background water. So this was early data for us. And then, of course, we start asking why and so what? And that's kind of where our group is now. One of the next things we could do with these species lists is ask, well, who is there? Are there human pathogens, fish pathogens? We also could ask whether there were any of those conspicuous harmful algal bloom members that we, or entities that we think a lot about in the Great Lakes and small inland lakes in Michigan. And that's what you can see here. So along the bottom, you have those three biomes again. This is the two types of background sampling, and on the right axis is the plastic communities. And when you see higher levels in the along the x-axis and these points, that means that those genera, so these are in these boxes, different microbes that the World Health Organization has identified as human pathogens. And for some classes of these microbes, they have been identified at higher relative abundances on plastics, and in particular on plastic near wastewater treatment plants and urban sites. I always feel obligated to share one caveat in interpreting this, which is that we also know that pathogens, human pathogens, tend to be biofilm formers, and we know that microbes living on plastic have to be biofilm formers. And so we are still trying to tease apart what this means in terms of implications for human or animal health, but these are the trends that we and others observe. Okay, so through with our next take home, which is that the microbes that live on plastic are distinct assemblages, and we're looking now into the potential microplastic interactions that could arise from these distinct assemblages. The things that we think about are the effects of the plastic, as well as the plastic leachates. So the chemicals that are coming off of the plastic, whether from dissolved molecules of the polymer or other additives that are included during the production of the plastic. What are the effects of those molecules on the microbial community composition? So essentially that species list, but also the function of those microbes. So what are they doing in this environmental ecosystem context, right? We did talk about how microbes matter based on the aquatic food web. If we are introducing this novel substrate with organic molecules that we know microbes are well suited to be either degrade, so assimilate into their cells, or can be negatively affected by, what are the consequences? And that kind of leads into the next effect. So switch it around, the effect of microbial biofilms on the plastic fate, either through degradation, but also the impact of biofilm growth on what that plastic particle is doing in the water. So we have some really excellent hydrodynamic models that are able to predict the fate of plastic in aquatic systems. Where is it going to go, at what rate? But very few of those are able to integrate, and I argue none at this point realistically are able to integrate the effects of biofouling. So the effects of biofilm growth on how that influences the way that plastic moves through these aquatic systems. And some visuals of what we're doing in the lab to begin to tease apart these microplastic interactions. So this is a summary of the potential outcomes of microbial enzymatic activity that we think about. One is the biodeterioration. So this, I should have put a citation. I forgot. I will add it before we send the slides around. These are not my words. But in this first depiction, we have microbes that are living in biofilms are really quite well equipped for the production of extracellular enzymes, which can then go about their enzymatic attack of the polymer surface, which can lead to biofragmentation, so breaking up the plastic into smaller pieces, which may lead to assimilation, which would then mean the picking those smaller molecules back up and bringing them into the bacterial cell or microbial cell. And then one final outcome of that would be the mineralization of some of those molecules back into their inorganic form. So turning the plastic ball, carbon of the plastic polymer into things like CO2 or methane. So those are the possible ways that microbial attack of the polymer can lead to different fates for the original carbon that comprised that polymer. So it's not just biodegradation, it's many different fates within that potential. And so how do we study that? Well, we work studying the microbial activity in situ by building things like these very simple in concept deployments that take a lot of effort to build. So this was our COVID full lab effort full lab plus dogs and chickens and kids and friends when we were trying to save our experiments from being lost last summer. And where these then were sent, where they were built into these vertical arrays and were sent into two different lakes, one up in Ontario and one up in northern Michigan, where they were, they still are actually. So they were sent out for a few months, up to 15 months, and we're able to retrieve them and study things like the rate of biofilm growth, the impact of the exposure, environmental exposure on the quality of the plastic, and able to tease apart things like depth and UV exposure and whatnot. So we're able to put them out into these lovely lakes, especially when you're able to collaborate with wonderful people. This is a multi-team effort led by Chelsea Rockman at the University of Toronto. And so these are the ELA lakes that we are putting some of our deployments out in. Another goes up into the University of Michigan's biological station, and this is what it looks like when the teams are retrieving those. This kind of gives you a visual impression of how depth can influence the rate of biofilm growth and formation. And we bring them back into the lab and send a whole suite of biological tools for biological inquiry to them, and also team up with a lab in the chemistry department who does a lot of interesting spectroscopy and other characterization of the plastic surfaces. So this is all work in progress. We also study more directly the impacts of microbial activity in the lab by studying there are hundreds of published studies of either bacteria or fungi that are able to degrade different forms of plastic. And so we've been accumulating and building a library of cultures in our labs so that we can more explicitly study the different reactions that they're able to perform and try to tease apart the different degradation mechanisms and rates under different conditions that these microbes are able to carry out. And then zooming back out to this potential, lost a bit of text there on the right. I will fix that for you too. Potential ecosystem level impacts. I did already touch upon thinking about the impacts of biofouling and how the growth of biofilms on plastic particles, Sam mentioned, are attempts at trying to better predict, model, track where plastic is going once it reaches the environment. These types of, I guess, physical implications of the biofilm growth, both from the biofilm itself through biofouling, but also through those other degradative processes through fragmentation or other forms of degradation, can impact where the plastic falls in the water column, and then where it falls will then have implications for the functions of the microbes that comprise those biofilms, in terms of what the impacts might be on, for instance, carbon cycling. Are we going to have more plastic up in the surface of some regions? That could be a home to things that are doing photosynthesis and primary production, or if the majority of plastic ends up being down in the hypolimnium where there's less water, maybe we have other microbial processes that are going to be kind of favored on these plastics. And so trying to tease apart where the plastic's going, what are the microbial processes that may be affected both within the biofilm of the plastic as well as in the broader water column, are important next steps for us as a community to think about. And this is just a flavor where we're going and how we're trying to piece together these different thoughts. This was just a nice visual of the experiment up at ELA that's going on, which actually involves these different mesocosms, limnocorals in the lake, where plastic of different polymer types has been added, where the fate through time can be tracked in terms of where the plastic is going and how different ecosystem level processes are being affected. And that's that. Happy to transition into questions.
Excellent. Okay. Getting back here. All right, great. Great. Thank you both. Okay. We will turn now with a good amount of time left for questions to questions. So, and just a reminder, yes, to take a look at the questions that are currently submitted and upvote them if you'd like, or continue to submit ones of your own. So I will start first with a question from Izzy, and this was during your presentation, Sam. What kind of pollutants are becoming airborne as a result of plastic incineration?
Yeah, that's a good question, and I don't know that we know the full answer to that. But many plastics contain chlorine, and the point of that is that when you're burning complex hydrocarbon-based materials and there's chlorine present, you form molecules like dioxins, polychlorinated biphenyls, PCBs. And these chemicals are known, well, dioxins and furans are the most toxic man-made substance. So, and they are generally formed in combustion type processes. So those are likely to be coming off of the incineration of plastics and similar type compounds. So you typically are going to get ringed chlorinated type molecules that have known human health impacts. So they're carcinogens, they're mutagens. So those are the types of, in addition to, of course, you're going to get CO2 and CO and methane and, formaldehyde and the kind of the whole suite. But the ones that you would be most concerned about would be those dioxins, furans, the PCBs that are likely also to be coming off. Industry will, of course, point as they will say that they're not going to be produced in very high concentrations, but these things start having an impact on organisms at the part per trillion level. So it doesn't really take them being released at very high concentrations for them to be having a very important impact on the ecosystem, and humans included.
Mm-hmm. Great, thank you. Mm-hmm. Staying in that contaminant, this sort of other pollutant questions, Teresa asks, has there been an evaluation of PFOS as a result of microplastic presence? And I don't know, I mean, this might be a question that both of you would have input on.
I was on a panel discussion with so our lab has not, and I don't know if Melissa's has or if she knows people who have, but there's PFOS does adsorb to the surface of plastics. And so that has been shown. And so in waters, this is always one of the concerns with plastic, is not just the chemicals that are in plastic, but the chemicals that adsorb to the outside of plastic. And then when those particles are ingested, and microplastics are very easy to ingest because of their small size, it brings those chemicals into organisms. And so PFOS have been shown to adsorb onto the surface of plastic. So if you're talking about a river that has known PFOS and undoubtedly also has microplastics, and those two things are definitely connected.
Mm-hmm. Thank you. Melissa, anything to add or? No,
just
okay. All right. we'll have to look for more information on that question. Moving now, following this human health thread from Deb, a question, any information on how microplastics in human consumables affect human health?
So that is really kind of the forefront of the research on microplastics. I took those slides out, but I mean, there have been some really interesting studies, one showing that it's in fecal matter. So we know that humans are ingesting it, but I think the more interesting studies and kind of disturbing studies are ones that have shown that there's microplastics on both sides of the placental boundary after childbirth. So there's no doubt that it's making its way across the placental boundary into a fetus during gestation. And a really interesting study that not only looked at fecal matter of adults, but paired it with the fecal matter of their newborn children, found 10 times as much microplastics within the fecal matter of the children as compared to the adults, which in many ways makes sense because children, they chew on plastic binkies, they crawl on plastic carpets, they play with plastic toys, they're drinking, formula out of plastic nipples and bottles. And so they tend to get a higher exposure to microplastics even than adults. And so there's a lot of concern about what that means, but I'm not sure that we really have any good data yet. And the data is always going to be difficult to tease that apart. If we're already exposed to microplastics before we're even born, and then at the age of 40, we come down with breast cancer, how are you going to know that it was because you were exposed to microplastics as opposed to pesticides in the food you ate or, something that you breathed in? It's really difficult to kind of tease those kind of systemic problems apart. So there's always going to be doubt that can be cast on any kind of results that come out of laboratory studies. But I do know that that's kind of the forefront, is trying to understand the human health implications of the presence of microplastics. Mm-hmm. Melissa, maybe, I don't know, you're the biologist. I should let you answer that more than me.
It's a different scale when we think about biology. The things that come to mind are not human health, but I could see it as a model for studying human health are, looking at in terms of human microbiome impacts. The interaction with the microbes could be, but I think for that we need more of a better understanding of the numbers that we're ingesting and their fate within the body accumulation.
So food for future research from both your labs. There you go. Christopher.
Not our labs, but other labs, yes.
So Christopher has a question about, he said he noticed the concentrations are being reported in your presentation, Sam, by surface area. Does this mean that you alluded to this, I think, but just making sure he understands, does this mean that the majority of particles lie on the surface of the water in this case, or not?
Yeah, so in our manta trawl studies, it is common practice that because the manta trawl only goes about 8 centimeters in depth, that they're reported as surface concentration, so units per surface area. In our river studies, because we do, and we have river studies that actually look at depth, but so in our river studies, we actually report them as concentration, so per unit volume as opposed to surface area. So the one graph where I showed the river data, all of the Great Lakes data had been put into concentrations as opposed to surface units. And so that was a pure concentration. In terms of the question, so the majority plastics generally, virgin plastics, I should say, either float or sink. They're either less dense than water or they're more dense than water. And so they tend to bifurcate that way. In addition, but as plastic is in the environment, well, when it's manufactured, chemicals are added to it that can increase the density. As plastic is out in the environment, it builds up bioforms. That's where Melissa is talking about these, and those biofilms will increase the density. So plastics will, as they exist in the environment, tend to increase in density and eventually sink. So kind of the sediment layer is the ultimate kind of sink, as we say in science, of microplastics. So we tend to see higher concentrations within the sediment than we do within the surface. You can actually see microplastics kind of moving down the column, but you tend to see higher concentrations in the surface or at the bottom, as opposed to the water column itself. But you definitely have concentrations moving down the water column because of that kind of density separation that can happen. But they tend to be, largely centered either in the surface or the sediment. That was a very long-winded answer, but hopefully the information was helpful and interesting.
Definitely expanded. Yep. Thank you. Gregory's asking, are there also microfibers of cotton and wool? And if so, do they cause similar issues?
I've been talking a lot. Melissa, do you want to take a first stab at this?
Yeah, I mean, this is definitely tangential to what we do, but I can say yes. So and probably also a bit speculative, but what I could answer as a microbiologist is that cotton and wool are, well, I don't know, actually, cotton. So from what else I've learned of the textile industry, cotton and wool, they do tend to also be coated by substances that help them be more robust or perform their job in textiles better. So I don't know what that does in terms of their resistance to degradation, but when we think of other fibers, biogenic, wool and cotton are biogenic. We think of things like the fibrous material in the plant material that we find floating, where we also collect microplastics, or like cellulosic material in the sediment. We know that the microbes, be they fungi or bacteria, archaea that have kind of co-evolved with this substrate, have had the evolutionary time to evolve the enzymes needed to break them down and to kind of assimilate and eventually mineralize those molecules. So when I think of problem, I think of it in terms of like fate, long-term fate. How long is this human-made material going to be in the environment? I'm not sure what problem was being referred to here. That's how I think of it. So yeah, if I think of cotton and wool, I think their potential for degradation is going to be higher, biodegradation. I don't know what the effects of those coatings might be on the potential for microbes to access the core wool and cotton. Also would then ask, what are those coatings, and do we have the same type of organic pollutant leachate issues with these other microfibers? So that's my off-the-cuff spin of that question. I'm going to toss it back to Sam, who I think thinks about this a little bit more explicitly.
I think microfibers they've become the most ubiquitous, although in the Great Lakes, the lower lakes, Lake Erie and Lake Ontario, we don't find as many fibers as we do up in Lake Superior, Lake Huron and Lake Michigan, especially Lake Superior. I think somebody had asked about the sources of microplastics in Lake Superior. It is largely fibers. They tend to be the dominant microplastic that we find up in Lake Superior. And so we think that the main kind of source of them is coming actually from contact with air. So it's coming from deposition. And the large surface area of Lake Superior helps to explain why you have then so many of them, because of that air-water interaction. So we don't tend to find as many of them, and kind of urban environments as we do in kind of maybe environments that are less, they tend to be a greater percentage in the more rural kind of environments. Sorry, I'm getting off topic. So my point is that I think in terms of our laboratory, because of the way that we process our samples, I think a lot of the natural materials tend to be degraded through our wet peroxide oxidation. When we do analysis verification that the particles that we're retrieving are in fact plastics using FTIR, most of the time they are in fact verified to be plastic. We have very few kind of like false positives or things that we picked out. Even cellulosic materials are oftentimes manipulated, and Melissa was kind of referring to this, in order to increase their durability. And so they're almost in that semi-synthetic category as opposed to fully kind of natural materials. And so so those are the ones that we tended to find. But in terms of their impact, it's probably a scale. And so somewhere in between. I don't know if that was helpful. I think, Melissa, you were better speaking in that question.
Nope, those are good answers. Thank you. Okay, the next question, which I thought where is it?
Maybe when I typed an answer, it went to answered. I think it was about degradation rate model.
Yes, thank you. If you could
I just
answer
put a
that,
citation.
right?
Yeah, I just put a citation for most recent study that comes to mind that we talk about in our lab, that has set the estimate to be tens to hundreds of years.
Tens to hundreds of years.
Yeah, but rate. So I was just looking more closely at the phrasing of the question. Oh, and rate. Sorry, and that's microns per year in terms of rate. That was published in that study.
Great. Okay, let's go to Margot's question about get to a solutions related question. Are there things that wastewater treatment plants can do to remove or reduce plastics?
I personally don't think that this is a wastewater treatment plant operator's issue. Like I actually, what studies have shown is that wastewater, and I should have mentioned this, wastewater treatment plants have actually been shown to be surprisingly effective at removing microplastics, somewhere between 75% and 95% effective on the removal. Still, we were getting the 4 million coming out from each facility on average, from an average facility on an average day. But they're pretty good, even though that wasn't their intention. So my favoring when it comes to solutions for microplastics are not at that point. It's upstream from that. It's looking at, and it's really kind of looking at the very beginning of this, our use of this, our production of this material, our manufacturing of this material, product design, those kinds of things, I think are much more cost effective and better ecologically than thinking about how wastewater treatment plant operators can do what they can do. It's not their issue. It's an our issue. It's a product design issue. And we didn't have a chance to mention this, but this is definitely a huge environmental justice issue. The vast majority of these facilities that ultimately produce plastic materials are centered in economically depressed areas and areas with people of color. So that's where the problem needs to be solved, is there.
Thank you.
And quickly spin off of that. It's very it's a spin. But your comment, Sam, on the light that this shines on environmental justice reminds me of just how I've also come to relate to the plastic problem, is that it is really a diagnostics for so many of our societal ills. I feel like I remember one of the early talks that I kept seeing presented from the plastic industry when they would come to our meetings was talking about the benefits of plastic for preserving a cucumber. It's talking about, how many days longer the cucumber can last in the grocery store, in your fridge. And I'm thinking about this in like January, February. Here we are with frozen ground. Should we be eating cucumbers? And to me, that was screaming about like food systems, local food systems, and bringing back the value of eating seasonally and making that accessible to folks that do want to live that way, rather than having folks incur the burden of then needing to manage the waste that comes from their plastic covered cucumber. So I think that's been a useful kind of thing to recognize and to communicate with the communities that I interact with in terms of the plastic story and the plastic problem. And it's not just this basic research, it's also an interesting element to reflect on in terms of our behavior as a society and individuals.
Yeah, exactly.
So whether we're working at a very micro scale or at a macro scale, the ability to zoom in or out and think about systems is an important part of the solution here. Yep, very good. We're going to try to just plow through a few more of these questions here, maybe a little lightning round before we end.
We'll be short. We can do this.
Yeah, no, it's good. No, thank you so much for your answers so far. So Mary asks, is there a correlation between plastics quantity and the number of public recreation facilities along freshwater sources in the Great Lakes? I don't know, right, circulation wise, if that makes much of a difference.
On beaches, there is. There's definitely on beaches. When you have high recreation rates on beaches, you see more debris.
I'd say at the basin level, it's more strongly correlated with population density. So cities.
Okay, great. Thank you. Diane, hello Diane, asks how much of plastic and tap water comes from plastic in the water source versus the PVC pipes transporting the water. Any ideas there?
I think honestly, it has more to do with it. It's the water in contact with air. I should have said this too. Water treatment operators, you are doing your job very well. We here in the United States filter our water. We have, by and large, much better infrastructure than most places. And so they do their job well. But again, as soon as that water is in contact with the air, it's in the air, it's in the water. And so I think that's where it's coming from, not so much the pipes.
Okay, thank you. Let's see. Julie asks, do you know the origin of these plastics? Anything else to say about the origin of the plastics? Is it primarily consumer goods, or are there other big sources we as consumers can try to take action on to reduce the presence of plastics in the environment?
Every plastic item you use will end up leaching plastics into the environment. So that whatever you can do to reduce your plastic usage has a difference. So I always encourage people to take the simple steps, no plastic bags, no bottled water, changing, I get my shampoo in stainless steel. Just all of my cleaning products are in glass. You find ways to, I get my toothpaste as tablets. It's just like every single thing, buying cotton and wool, hemp.
Great. Okay, one more question from Jane for today's time together. Is there a quick and easy way, quote, quick and easy way to assess microplastics in a water body, lakes specifically, she's asking about? No would be the best answer.
Melissa, do you have a yes answer?
Yeah, quick and dirty. I took out my slides, my COI slides about quantification and how not quick and dirty. Well, dirty, yes, but quick. Yeah, no, I don't have a quick answer other than that is what caused us to step back from researching this problem about 6 or 7 years ago, because we are a microbiology lab, not chemistry, and we thought, surely in the next 5 or 6 years, this is going to be all like high throughput quantification, spectroscopy based, like send your water through a flow and it's going to get the spectra to confirm, like synthetic polymer from natural biogenic polymer. It's really, really hard and takes a lot of tedious manual effort to, if you really want to get a good count of micro scale plastics, or the smallest, which are the most abundant. But there are ways. I know a lot of citizen scientist initiatives that are collecting and counting plastics. What do you have to add?
Bigger plastics, yeah, not micro. Like, focus in on the big stuff, and that gives you an idea of the small stuff. You're going to find more small stuff than big stuff.
Well, Jane, sorry we didn't have a happier answer for you there, except yes, there are lots of citizen scientists out there helping to do the assessment that they can. We call this our speed networking webinar series, so you could actually meet Sam and meet Melissa and hear a little bit about the work that they're doing. And we hope for those of you that we did not answer your questions that you would follow up with Sam and Melissa. They've agreed to do some of that, and we're grateful for their presentations today and grateful for their willingness to have a few follow-ups with you all. So you can see their emails here. Thank you both, Sam and Melissa, so much for great presentations, and thanks to all of you for participating and adding your voices to the conversation today. I know on these webinars, sometimes it can be just a sort of a passive intake of information, and we love it that you're participating and contributing your expertise to the conversation. So thank you so much for your time today, and we wish you a great rest of your day and a happy 2022.